Okay, we're all set.
All
All right.
right.
I'd like to thank everyone for coming. First and foremost, my name is Kurt Farrell. I'm at Mount Sinai. I'd like to thank the organizers of this particular seminar, including Hannah at the NACC, as well as Dr. Duggar, also Dr. Murray, and Dr. Flanagan, who started this and have given me the ability to kind of take it over and let you hear from different speakers working in the area of digital pathology. That being said, I just want to give an open call to anyone who wants to speak at this particular lecture. Please send me an email. I will put my email in the chat. We'd love to have you. And so today we have close friend of mine. I'm really happy that she's able to speak today. Sheina Emrani. She completed her doctorate studies at Rowan University in clinical psychology. Then she went on to Brown to do postdoctoral fellowship, and then now is in a second postdoctoral fellowship with Dave Irwin at the University of Pennsylvania in their Penn Digital Neuropathology Lab. She and I met a few years back at the American Association of Neuropathologists annual meeting. We struck it off. We had a mutual interest in progressive supranuclear palsy, a rare neurodegenerative disorder, and kind of the neuroinflammatory factors around it. And I think some of her talk today will go over that. So I'm just really excited to hear about all the amazing things she's been doing in terms of research. So today she's going to be talking about some of the unpublished current things that she's done. So with that, take it away, Sheina.
Thanks, Kurt. That was really nice. So again, I'm Sheina. I'm a postdoc in the Irwin lab. I'm also a neuropsychologist by training, and I'm really excited to talk about digital pathology and how that relates to cognitive outcomes. And so today we'll talk about neuroinflammation as a contributor to global cognitive dysfunction in FTLD-Tau. I have no disclosures. So just a brief primer in terms of the nomenclature when we talk about FTD and FTLD. So FTD, or frontotemporal dementia, describes the clinical syndromes that we see in life, and those span across behavioral variant FTD, PPAs, primary progressive aphasias, all the way to progressive supranuclear palsy, or PSP, as Kurt had mentioned. Each of these clinical syndromes can have specific underlying neuropathology that we call frontotemporal lobar degeneration. So now when we talk about FTLD, what we're really describing here is the underlying pathology. And one thing to note is that these clinical FTD syndromes, you can see that they're color-coded according to the proportion associated with a specific pathology. The two most common forms are FTLD-Tau, which we'll talk about today, and FTLD-TDP. One part of FTLD that we know is that it's been linked to neuroinflammation. It's really important. The FTLD pathologic process is important and linked to neuroinflammation. When we talk about neuroinflammation, there are two main types that we're typically describing, although there are more. One are these microglia, and the other is astrocytes. Their main responsibility is to make sure that the neurons are happy and things are working appropriately. In the acute phase, you would expect to see some neuroinflammation, but what tends to happen is with these pathologic processes of FTLD, you end up having chronic neuroinflammation, which is not good long-term. It can ultimately lead to neurodegeneration and worse cognitive outcomes. In this review by Bright, they also describe what we know so far when it comes to FTLD. A lot of studies have been done looking at these genetic mutations, and in addition to that, most studies have really looked at FTLD-TDP, less so with FTLD-Tau. One study from our group showcased how FTLD-Tau is related to gliosis. And so what you can see here on the left-hand side is this high-resolution MRI, a T2 star weighted image that's able to capture iron accumulation. So these dense bands that you see with these green arrows are showing iron accumulation in a brain who came to autopsy and had FTLD-Tau. If you look at that middle image, you can see this mid-layer iron-rich band that corresponds to what we see on the MRI. And then looking at it microscopically, what you can also see is these morphologies that look like these activated microglia and have iron accumulation, showing again this relationship between neuroinflammation in FTLD-Tau. There have also been in vivo studies looking at the association between neuroinflammation and cognitive decline. So one study used ligand TSPO, which typically is a measure of neuroinflammation, specifically activated microglia. And what they show in the frontal lobes of people who had the FTD syndrome, those who had higher levels of this TSPO, so suggestive of worse neuroinflammation, ended up having a steeper cognitive decline over a 5-year span, showing that there's this association too with neuroinflammation and cognitive decline in life. One recent paper by Kurt that y'all just heard about briefly with PSP is that there is a clear link between genetics and the importance of glial cells in PSP, or progressive supranuclear palsy, with neuroinflammation and other neuroimmune genetic risk alleles as well. And to further this, Kurt and Adam Nash, who's here at Penn, showed that there's this new risk gene C4A. And what you can see here is this co-localization of C4A with hyperphosphorylated tau in these oligodendrocytes, which is another aspect of these glial cells, with these positive coiled bodies, again, showing the importance of neuroinflammation in FTLD. And this has extended into other neurodegenerative diseases as well. So a really nice paper that also came out in 2024 by Naomi Corey, Melissa Murray in the Mayo Clinic. They found that there's this complex relationship between tau and glial markers when you look at people who have hippocampal sparing AD, typical AD, or limbic predominant AD. And what you can see here on the lower band is both astrocytes using GFAB and these activated microglia and macrophages with CD68, and its landscape across frontal, parietal, temporal regions, and as it relates to tau pathology again. And finally, another wonderful study that came out this year by Olivia Capasi, Julie Schneider at Rush, showed again that when looking at reactive microglia, specifically in the hippocampus, it does have a direct effect on cognitive decline. So aspects including episodic memory, semantic memory, processing speed. And they showed that microglia burden was associated with faster decline above and beyond the presence of these common pathologies, neurodegenerative and cerebrovascular pathologies, again, demonstrating that neuroinflammation should have a seat at the table when it comes to these primary pathologies, and that it might additionally contribute to our clinical endpoints as well. And so we know that there's this relationship between FTLD-Tau and neuroinflammation, but it's not entirely clear as it pertains to FTLD how that relates to our cognitive outcomes. And so that's exactly what we sought to do. So our objective here was to investigate the relationship between these post-mortem markers of neuroinflammation and tau pathology with cognitive dysfunction, specifically in FTLD-Tau. And our hypothesis was that neuroinflammation would have a unique contribution to cognitive impairment beyond tau pathology. Again, as I mentioned, I'm a neuropsychologist, and what's really exciting is the ability to use these digital pathology methods to measure density of pathology that will enable us to facilitate better statistical modeling so that we can account for more of the variance when we think about these cognitive outcomes. And I think that's super exciting to do. So for this preliminary study, we first surveyed our database, INDD, and we found people who had the FTD syndrome in life and who had neuropathologically confirmed FTLD-Tau. After that, we wanted to ensure that there was some sort of cognitive measure in life. So we utilized the mini-mental as our global cognitive outcome. And fortunately for us, there was a subset of individuals who had more robust neuropsych measures. In terms of our regions of interest, we looked primarily at 6 different areas, 6 different brain regions that are implicated in FTLD-Tau in terms of tau propagation and acceleration. And what we did is we annotated the gray matter along the sulcal wall, and we quantified a percent area occupied. So based off of our pathologic measures, we were able to capture how much pathology there was along this sulcal wall. And for this study, we actually annotated over 2,000 slides. I, coming into this, had less experience with digital pathology, how much work is involved with annotating so many slides. And it really highlights how fantastic it is to have a wonderful team in the Irwin lab to be able to do this together. This is a huge feat, and I'm very appreciative that we were all able to capture so many slides and be able to annotate them. In terms of our pathology markers that we quantified digitally, we had AT8 for tau. We had GFAP for these reactive astrocytes. And finally, we had ferritin for reactive or dystrophic microglia. Ferritin historically was used to be able to capture these dystrophic microglia until HLA-DR and CD68 came on board. And while ferritin does a really good job at primarily capturing these reactive or dystrophic microglia, to some degree they are also seen in neurons as well as astrocytes in terms of this iron accumulation. And there is a subset that you'll also see some oligodendrocytes. But for the most part, ferritin, which is this iron stain, does tend to capture these dystrophic microglia. With our global cognitive measures, we focus primarily on the mini mental state exam. So that's a purely cognitive measure out of 30 points. The next, we also were able to use a subset of individuals who had the clinical dementia rating scale. So now this is not only looking at cognitive abilities, but also functional abilities as well. So Informant would provide us with additional information in terms of how they're doing with their day-to-day functioning. So this is both cognitive and functional. And finally, we used another task, confrontation naming, where people are shown a picture and are asked to say what that picture is. And fortunately for us, because of NACC, we were able to utilize the crosswalk for people who had Boston Naming Test or the MINT, both of which are these confrontation naming tasks. And briefly, in terms of our clinical characteristics, you can see the percentage of each FTD syndrome. So we had cortical basal syndrome, behavioral variant FTD, non-fluent PPA, and progressive supranuclear palsy. And we had a good display of both males and females across these groups. And so as the starting point for our preliminary analysis, we wanted to ensure that what we were seeing in terms of ferritin was truly pathologic. So what we did here is we took our FTLD group across those core regions that I'd previously described, and also a normal group. So people who came to autopsy who had an unremarkable brain, pathological aging, or part, to determine whether or not what we're capturing with our percent area occupied for ferritin that should measure this dystrophic microglia is in fact pathological. And what you can see here is that there is a statistically significant difference between normals and FTLD across all the brain regions, but additionally in the middle frontal cortex and orbital frontal cortex, suggesting to us that this increase is truly pathological. We also wanted to do the same thing with GFAB, or so these reactive astrocytes to ensure that this isn't just an age-related phenomenon, although individuals with FTLD are typically younger. And what you can see here again is that the percent area occupied is statistically significant and higher in our FTLD group, suggesting again that this is truly pathological. And so to start, we looked at the correlation between our markers, so ferritin, GFAP, and AT8, and you can see a mild to moderate correlation, which is something that we would expect. And importantly, they're not so highly correlated that this would be problematic when we put them into models for multicollinearity purposes. So this was a good check to us that we can go ahead and move forward with our analyses. So to get into the meat of this study, first we looked at the mini-mental state exam, tau, and neuroinflammation. So one thing to note is people who had a score of 30 were suggested to be cognitively intact. Lower scores are suggestive of cognitive impairment. And we averaged across all the brain regions to first see if there was an association between the mini-mental and our markers of choice. And what you can see here with this bivariate correlation is that they're each independently associated with the mini-mental in the direction that we would expect. We then asked the question if neuroinflammation would provide additional information above and beyond tau pathology, so using this AT8. And the way in which we did this is using a nested model. So in our first model, we just included tau pathology and our covariates. And in the second model, we included not only tau, but ferritin and GFAP, suggestive of these reactive astrocytes and microglia, and asked the question if this would improve our model fit using ANANOVA. And we see statistically significant results suggesting that neuroinflammation does provide additional information of who is more likely to show cognitive impairment above and beyond tau pathology. We wanted to see if we can replicate this using another global cognitive measure. And so we used the Clinical Dementia Rating Scale Sum of Boxes. So now higher numbers are reflective of worse cognitive outcomes. And again, you can see the relationship here where they're each individually correlated with the CDR Sum of Boxes in the direction that we would expect. We ran the same exact model, and again, we do see statistically significant results suggesting that neuroinflammation does additionally contribute to cognitive impairment above and beyond tau pathology alone. We wanted to tease this apart a bit further, and so we ran a mediation model. And so one thing to note here is that we almost have a partial effect here. So to highlight that it's at 0.06, but importantly, this relationship here of tau pathology to our mini-mental performance still remains statistically significant so that it would only be a partial mediation. However, when we look at the same mediation model, but with ferritin, what we now see is a total mediation. So there's this attenuation between tau pathology and mini-mental performance. And so we're currently increasing our sample size to see if this holds with larger numbers, which is something that's important for doing a mediation model. But I think this is helpful in suggesting that we need to move forward in understanding what this relationship is, and that perhaps neuroinflammation should have a seat at the table when we're thinking about cognitive performance and cognitive decline. So this data is pretty encouraging. We also wanted to determine whether or not there was some sort of specificity with our findings. And so now we restricted our brain regions to those that are needed for something like confrontation naming, including the superior middle temporal cortex, entorhinal and parahippocampal gyrus, and the angular gyrus. And again, higher numbers are reflective of better naming performance. And you can see this relationship across these two markers. And running that same exact model that I had previously described, we again see that when you include neuroinflammation into the model, it improves our model fit and helps us to better capture and better understand the reasons for why someone is experiencing not only just global cognitive impairment, but now we have some sort of specificity with the brain regions that are associated with a specific task or a specific cognitive domain. And importantly, when we ran the same when we looked at a correlation with an area that's less involved when it comes to naming. So the middle frontal cortex, we don't see that same association, suggesting again that this is likely specific to those brain regions and respective tasks or cognitive domains. And so to summarize, post-mortem neuroinflammation is related to global cognitive impairment, nearest death, in FTLD-Tau. And when you include these neuroinflammatory markers into a model, it improves fit, suggesting that they do additionally contribute to our understanding and cognitive impairment, who is more likely to have some sort of cognitive impairment. We do see this specificity with temporal regions and naming, which I think is also, again, very encouraging, especially if we want to really understand these clinical endpoints and what maybe potentially should be included as we're trying to understand these things in people who are living. And I don't need to necessarily say this to this group, but digital histopathology metrics are really important in helping us improve the clinical pathologic correlations in FTLD or other neurodegenerative diseases. And I think this is super fantastic and looking forward to learning more about digital path. There's still a lot of things that we don't know and that we're learning. One study by a wonderful assistant professor here at Penn, Katie Cousins, wanted to interrogate whether or not, if there's a way that we can, using plasma GFAP, which we have, which is a measure in life, correlates with what we see in the brain as well, used in both FTLD-Tau and FTLD-TDP. And from her study, you can see that there is this correlation between plasma GFAP and brain GFAP percent area occupied, which is, again, very encouraging if we really want to understand how we can best help people in life and how these plasma biomarkers correspond to what's occurring in the brain. And so future work similarly will include these ante-mortem measures of neuroinflammation, neurodegeneration to extend our observations longitudinally during life. And I just wanted to thank the co-directors of the FTD Center, especially Dave Irwin, who's been a fantastic mentor. Everyone in the Penn Digital Neuropathology Lab, I couldn't have done this without them. The ADRC here, and for the participants who graciously donated their brains. Thanks.
That was amazing. On a huge effort there, a lot of work to analyze all those images, annotate them. I have a lot of questions myself, but there are a few in the chat to start, so I will get to those first. So first question says, great work. Have you looked at phosphorylated tau? And does it have a similar correlation with total tau pathology?
So we looked at for our post-mortem neuroinflammatory markers, we only looked at AT8. We haven't necessarily looked at other markers of tau pathology. This is an ongoing investigation so far. And from the biomarker perspective, we haven't looked at that yet either.
Right. And the next question starts out by thanking you. Nice presentation. If I mispronounce anyone's name here, I apologize. This is from Nafis. They ask, neuroinflammation is a dynamic phenomenon, and the relationship between microglia and astrocyte activation with tau may vary over time. Have you studied this time dependency? I assume that there are human studies indicating that neuroinflammation has a significant relationship with tau during a specific age window of 40 to 60 years, whereas this may not hold true for older individuals.
Yeah, that's a really fantastic question and something that I think we're constantly grappling with.
Isn't that kind of tough in the world of neuropathology, right?
100%.
Time point, which
100%. Yeah, I think that's a fantastic question. I think also from my understanding, and I'd be curious if other people have thoughts about this, is that when it comes to propagation, of astrocytes versus microglia. There's a difference there, whereas astrocytes seem to not propagate as much. They can to some degree, potentially through blood vessels, but once they're there and they stay in their domains, and they become reactive. Microglia are a whole different ballgame, I think, where they have capabilities of this propagation. And so looking at the temporal relationship over time, I would imagine it could also, and what comes first with microglia and astrocytes, I think that's a really important question, but something that's so challenging to do neuropathologically, and I think also probably in vivo at this point too, but I think that's a really important question and point.
Yeah, I think that some of the resources at the NACC, like some centers do collect CSF, so you could get a proxy for some of these things maybe in the living, and there are some new neuroimaging modalities that are coming out. Again,
Yeah.
probably stored within the NACC that people can utilize to answer that question. Great question. Moving on. Thank you for sharing great data. I'm Elise Masson, tenure track investigator at the NIH Center for AD and Related Dementia. I was wondering, have you compared your GFAP data with the pan-astrocyte marker line ALDH1L1? In addition, same question about microglia. Have you compared your ferritin results to the pan-microglia marker like IBA1? If
Yeah.
yes, what did you expect to see? If no, then what would you expect? Thank you.
Again, really fantastic question. We have not, we're actually currently working on that now, to do some of that work. In terms of what I would expect to see, I think one thing that's important here, that's potentially different again from the astrocytes and microglia, is that in the astrocyte world, they stay to their domain. When they die, from my understanding, there isn't as much propagation. And so one thing that I think would be really important is trying to quantify astrocytic degeneration, because when we use these digital pathology measures, we're getting the intensity or the DAB signal. But if we can use some sort of features to determine which ones are degenerating as it compares to this pan-astrocyte marker versus ones that are just reactive but not yet dying, I think is important. And so I would expect to see just a lot of death in these astrocytes. When it comes to microglia, we're also working on that now. I struggle a lot more, I would say, with microglia because I think there's a lot more complexity to it as compared to the astrocytes in terms of the type of activated microglia, what they mean, which are beneficial potentially and which are not. So I don't necessarily have a good answer, but we are currently working on this. So hopefully next round we'll have some information on this.
Awesome. Another great question. I'm going to read the next two questions together because they kind of coincide. And it actually was a question or a point that I wanted to bring up as well. So the first question is from Aaron Connolly. They say, thank you for such a great presentation. Would love to hear more about the annotation process. Annotating 2,000 images is a lot of work. How did you create a consistent process across the team of people doing the annotations?
Yeah.
And then the next question from Jerry Liu, this is an amazing presentation and study. Annotating 2,000 images is a huge feat. Would there be an interest need for creating a digital tool for annotations? If so, would the human annotations on 2,000 images be available to train a machine learning model? I was going to ask the same question because in my group, we are working on creating machine learning models to do the annotations for us. For me, we're trying to break down the hippocampus subfields. We have a tool that kind of works at doing that, but yeah, you need lots of training data to allow these machine learning models to learn the area to annotate. But it's difficult in brain because there's just subtle differences between regions. So I guess back to the actual question, how did you create the consistent process to do it?
Yeah, I think those are both fantastic questions. And so, Kurt, similar to what you were saying, I think a lot of people are trying to now develop some sort of automated annotation, which would be fantastic because this does really take a lot of time to do. So in terms of how we tried to make sure that we were doing it consistently, one is we always try to annotate along that sulcal wall, as I previously mentioned. That was one aspect of it. Two, when we're annotating, we also shut off the DAB signal, so we're not biased to how much signal there is actually there. So we're only looking at the hematoxylin at that point. To determine that. And then finally, I would go in and just check to make sure so we had some good inter-rater reliability, which we're also still improving and working on. So it's still definitely a work in progress, but those were the things that we tried to set so that we would ensure that the annotation process was as consistent as possible.
And I presume you were using some software like QPath or the
Yes,
image scope?
QPath.
You were
Yep.
using.
Yeah. So we did this all in QPath. And we set lines for the peel surface and the gray white matter junction as our way of annotating.
Fantastic. Yeah, we've been annotating in QPath. We've also been using the digital slide archive, which some people on the call are familiar with. I think there is an effort to answer the last question and not put words in your mouth, to make annotations available to those across the community. I think there's been a big push through the NIH to do that. Most of these come out as small JSON or XML files. So I guess that's ongoing efforts through the NIH to start creating repositories for us to upload these things so other people can use them, right? I don't want to speak for you, but I'd be happy to share my annotations. I just need a platform to do it.
Yeah, I mean, I think that would be fantastic for so many reasons. One, because this is a ton of work, and two, per the previous question, if we want to do some training models, we need a lot of data. And so the more data we have, the better. I will say it's really, I can't emphasize how much work it is. Again, as a neuropsychologist, not something that necessarily crossed my mind, and I'm really appreciative of the lab because it really is time intensive.
The next question kind of dovetails on this too. This is from Brittany Duggar. For Iterator with annotations, what metrics readouts did you use?
So we haven't done it yet. For now, it was just me kind of going through and checking the work. We have discussed ways of if we can use this to then be automated. But I think for the annotations, there are two things to consider, at least for some of the work that we were doing. One is to make sure that the tissue was not torn and that you had from layer one all the way up, which was one aspect of it that I think people in the lab are really trained to do. And the other is that peel the gray matter to white matter for the annotations. And so I would go in and double check that. But we are currently working on these inter-rater, but we don't necessarily have data to show for that yet.
So just really quickly to summarize what you just said. So you're the gold standard. Why I bring this up is so many times we're trying to figure out metrics. And usually when you do a metric, you have to have a gold standard to compare it to. But when you might have multiple raters and some might denote something as positive, denote something as negative, or not even denote it at all, that's one thing for the classification. But another thing is if you're doing tracings, right? So you have those two kind of things like segmentation versus classification and knowing what metrics to use, because this is an issue.
Yeah, you're absolutely right. Yeah, and I think we're constantly optimizing how we annotate too, which is great. But also it's a lot of work.
Yeah, absolutely. I've had some issues in my own work as well with that too. Not only with annotating regions, but annotating various forms of pathology, like what is a pretangle, what is an intercellular tangle, and there's disagreement even amongst highly trained neuropathologists. So definitely an issue in the field that have to get resolved.
Yeah.
Looks like there are no more questions in the chat. I just wanted to point out one final question that I personally had, which I thought was interesting. coming from a background of genetics, I think it'd be interesting to include that in your model as well, to see like maybe how the tau haplotype influences what's going on with the different pathologies and neuroinflammation, as well as cognitive outcomes. Because I know for the PSP GWAS that we did, you have most of the brains and I have the genetics on them. So I think that'd be another cool component to add to your already complex model that you're doing. That's not
Yeah,
really a question. That's just more of me thinking about how we can think about, what's going on in these postmortem brains. And, we have these psych variables. It's really cool to look at everything we can, including genetics.
yeah,
So we can
yeah.
talk offline about that. It looks like another question has popped up in the chat, or suggestion rather. This says, one suggestion, related PET neuroimaging, TSPO, is not a good target to study neuroinflammation. There are many limitations for this target as well as PET radio tracers targeting it. It's interesting to know.
Yeah, that's so good.
Not coming from, I don't have a strong background in neuroimaging, so it's always good to reach out to our colleagues that.
Certainly.
that
I think Sarah Dunlop also may have had a question.
Oh, yes. Sorry. Totally skipped that. Good call. Fantastic presentation. Have you observed a difference in microglial morphology within the ferritin positive population across your cases, or a spatial relationship with tau positive inclusions?
Yeah, also really great question. We haven't looked into that yet, but I think that is something that's really important that we also plan to do, and also using HLA-DR or CD60 with these tau markers, I think would be great too. Yeah, there's a lot of outstanding questions that I think are really important that we're just starting to work on.
Amazing. So with that, I just want to give myself a shout out one more time. We have such a large group here. At one point, there was over 100 participants. So again, if anyone wants to speak at this meeting, which is a really great way to share a publication you've published recently or one in the past related to digital pathology, or even a work in progress, we're really flexible in time and structure as well of what we're doing. So we just want to get ideas out into the community. So please send me an email. My email's in the chat, kurt.farrell@mssm.edu. And I appreciate everyone attending. And this was a fantastic discussion. Thank you so much for showing your amazing work.
Thank you. I'll also echo what Kurt just said. This was, it was a fantastic experience. So I recommend this to future speakers.
Amazing. Thank you so much.